in engineering sciences and material sciences, biotechnology and biomedical engineering. Now she joined Lumiere Datamatics as a research integrity associate, and there she has worked in maintaining the research integrity of published and unpublished articles, and was also briefly involved with the editorial board as an editorial assistant, where she reviewed around 1,000 research papers for their possible publications in reputed journals. Now she has joined JP Institute of Information Technology as a project research scientist under the project development and evaluation of rapid diagnostic test device for pediatric pulmonary tuberculosis based on detection of host and bacterial proteins in urine samples. She is involved in R&D for the development of biosensing platforms for urine antigen detection using lateral flow assays and electrochemical characterization. Thank you very much, ma'am, for joining us on this platform to deliver a talk on nanobiosciences and their measurement challenges, a 2026 scientific overview. It is very important for the medical sector, in the medical devices sector, as we are working in different medical devices. We have with us our Honorable Shri Biyandishit, our Director Legal Metrology, sir. He has done a lot in the field of legal metrology. Sir, thank you very much for joining us. I would like to request you to kindly speak 2 words, say 2 words, and inaugurate the workshop. Over to you, sir, please.
Very good morning, Sri Astos Agrawal ji, Director Legal Metrology, Government of India. Dr. Deepsika Yadav ji, today the chief guest of the program. Our colleague legal metrology officer from the different states. Regional Difference Standard Laboratory officers, ILM officers. Every day, Dr. Astos Agrawal is giving the very good challenging topic to the peoples. And he is bringing the good scientist also to speak on that very subject. That is very appreciable. People are getting day by day new challenges. So yesterday, just I got a thing for my next visit USA 2011. So that dream was completed by Dr. Asutosh Agrawal after achieving OIML CS certifications. And that was started during that time by me. Some point I remember that one kiosk I had seen at the NIST. That kiosk concept has not been cleared in India. Kiosk means that if you are entering into any organization, electronically like a big computer science, big computer reflection, you will find everything on that very thing. So you can just tape and go through the any section, any history, anything, any research and other type of things available in that very organizations and connect to those very divisions. So you can get a very good at present you will say that it is a website information, but it is not a website information. It is for the institutional organizations, informations and the facilities and so many other type of things that which is taking advance day by day in that very field. So that type of things still we could not develop in our countries. It is neither at National Physical Laboratory nor at any university or IT or AIMS I had seen there. So that is very important that those things is required for the development so that people may be aware about the nanotechnology and all this type of things. Because if the people will be not aware, then there will be less development for the uh, next generation people. So who is the most brilliant people coming to your organization? Is the youngest people who are coming first time to your office. Are some visitors who are coming from abroad and all that type of things. And there is nobody to guide them all the things there. So that QoS will develop a such things that you can get lot of information and knowledge from that very things and can understand lot of the things. So this nanotechnology is playing very important role in the food technology, in processed food technology, chemical technology, medicines, and so many other other wings also. So the people who are doing the research work, they knows very well. Think the topic is very attractive, and such type of things should be made available to the common people, so that the young people may be aware, and they should get the more encouragement from the organization, so that they can develop themselves up to some level. So from my point, this is the very very important to bring more talent in the country, and to bring the more curiosity among the talented people, so that our country may develop. With these very words, I inaugurate this very session. I give thanks to Dr. Ashutosh Agarwal ji, so that he can request Dr. Deepsika Yadav madam give her lecture, and the people may acknowledge that very lecture in a nice way. Thank you, Ashutosh ji. Thank
Thank you,
you.
sir. Thank you very much for your brief and wonderful inauguration. Thank you very much. We are all aware whether it is OIML, whether it is time dissemination, whether it is strengthening of the legal methodology in India. It is all credit goes to you. Under your leadership, we did a lot, and certainly under your leadership only we are trying to do better. Thank you very much, sir. Thank you. Yeah, I know you are always with us. Thank you very much, sir. Thank you. Now I would like to request Dr. Deepshika Yadav to kindly deliver her talk and enlighten us with her vast knowledge and experience. Madam, after this PPT, after this presentation, the video as well as this PPT will be available in the public domain. Thank you very much. Over to you, ma'am. Thank you.
Now, shall I begin now?
Yes, ma'am, please.
Hello.
Over to you, please.
Yeah. So, good morning to one and all present here. So, today we are here with another important and fresh topic, which is the nano biosensors and their measurement challenges, 2026 scientific overview. So, first off, let us discuss what exactly are nano biosensors. Nano biosensors are basically very important devices for us, and they work at the integration of the biological recognition elements, which in my last lecture I had talked about the different types of components of a biosensor. One very important topic, one very important component of the biosensor is the biological recognition element, or the bioreceptor, which is capable of detecting the target analyte, or the analyte of interest, or we can, in common biological terminology, we say that the ligand detection. So over here, nano biosensors basically are typical devices which are which are basically an integration of the biological sensing element, or the biorecognition element, along with vast majority of different types of nanomaterials. And these very nanomaterials are essential for providing a very ultra sensitive detection with a high sensitivity, which is a prerequisite for detecting the very low concentrations of the target analyte, or the biological molecule, which is present in the different disease specific scenarios. Now, why are they so important? Like, why these nanomaterials are being used in these sensors? That is why the name, that is nano plus biosensor. So nano refers to the nanomaterials which are used for the ultra sensitive detection, and biosensors are basically very important biological devices which are capable of converting a biochemical or a biological recognition event which occurs between the bioreceptor and the target analyte into a measurable electrical signal by means of transducers. So over here, the of detecting particular signal between the and a legal of interest. So why are they important? This is because they have very important properties which make them great candidates for being incorporated as nanobiosensors. So what are these properties? They can be a higher ratio, the quantum effects which occur, and the enhanced reactivity of nanoscale dimension. Now, the biosensors have a multitude of different applications in the healthcare, diagnostic, disease-specific, environmental monitoring, food safety, and other energy sectors. Excuse me. Nanobiosensors basically are, you can say that they are an amalgamation of the nanotechnology and biotechnology, which operate at the scale of the nanoscale, at the scale of individual molecules and viruses, which is related to the biotechnology area of the nanobiosensors. And these sophisticated devices basically leverage the unique optical, physicochemical properties of the different types of nanomaterials which and are engineered at the nanoscale dimension. So nanomaterials are basically those materials which have a size range from 1 to 100 nanometers, and this is which is capable of imparting the unique properties like the high surface to volume ratio, which dramatically helps in improving the charge transfer capabilities of the nanobiosensors, which is responsible high sensitivity, which is attained. Now, there have been many reports in the different reputed journals which are advocating the importance of these nanobiosensors for the detection of different types of diseases, as well as their application frontiers like medical diagnostics, environmental protection, and personalized healthcare. Now, these nanobiosensors, why they are so important? Because they have reported to attain limits up to zeptomolar level. So, attaining such low levels of detection make them ideal candidates for the diagnosis of diseases where the target molecule or the target pathogens or the target proteins are present at extremely low levels, which cannot be detected by the normal conventional methods. So, over here, nanobiosensors come to the rescue. Now, as I had already discussed in the previous slide, that nanomaterials are very important for nanobiosensors because they are basically like for a human body. When we talk about the human body, the cell is the basic structural and fundamental unit. Similarly, for the nanobiosensors, the nanomaterials are the basic structural and functional unit which imparts the unique properties which make them
Madam, your voice is not coming. Am I audible? Atulji?
Till that time, Dr. Saab, you can speak yourself on
Sir,
the nano measurement issues where
yes, sir. Thank you very
that
much, sir.
by using some
Sir,
very sophisticated balances where that what are the roles and all those type of things in detecting those very things because
sir.
the sensor plays the very important role, no?
True, sir. Absolutely right.
So you can start there till that time she is coming online.
So actually, there was some comment. This connection was broken. I think Dr. Deepshika Yadav has joined.
Okay, thank you.
Nice. Actually, I could not identify. I was continuously speaking, so I didn't know when the PPT stopped. So
Okay,
can
no
you
problem.
just let me know
You again have to share
from
the
where?
PPT.
Yeah, I will do that, sir.
RSL
So
Ahmedabad.
is the PPT presentation?
Yes, it is visible. At the same slide we were there, ma'am. Please, you continue
Uh,
at the same slide.
Okay. Okay. I was discussing the different types of nanomaterials, which are an important part of the nanobiosensors. And as I had discussed, let us begin with the metal nanoparticles. Why they are so important? Because metal nanoparticles like gold and silver, they have unique optical properties which are imparted whenever white light is incident on them. The outer electrons, they start oscillating in resonance with the frequency of the incident light, which imparts a very important characteristic feature of gold nanoparticles and silver nanoparticles, which is the surface plasmon resonance, which is harnessed in a number of different nanobiosensors nowadays. And these are in case of gold nanoparticles, the SPR phenomena is responsible for imparting the different colors that we see. And these colors are an important colorimetric detection platform, because as the size changes, the colors also change, and based on that, we can easily detect the target analyte. Now, nanomaterials are basically classified on the basis of how many dimensions are outside of the nanoscale. So they are basically divided into four types, which is the zero-dimensional material, one-dimensional materials, two-dimensional materials, and three-dimensional materials. So basically, zero-dimensional materials over here, all of the three dimensions are at the nanoscale. And in case of, for example, the quantum dots are one of the popular zero-dimensional materials. Then there is the one-dimensional material, where only one of the dimensions is outside of the nanoscale. Then there is the 2D material, which where dimensions are outside the nanoscales, and bulk powders, which we are commonly using for the precision of the nanoparticle. All of the three dimensions are large. They are outside of the nanoscale, which are powder. So for one-dimensional materials, carbon nanotube, one of the important and popular choice. And they have exceptional electrical conductivity and mechanical strength, which is responsible for their unique usage in the electrochemical types of biosensing. So basically, let us discuss another important nanomaterial, which is the graphene. Now, graphene is a two-dimensional type of nanomaterial. So for understanding this material, let us discuss basically is the difference between graphite, graphene, and graphene oxide. So for graphite is basically the bulk powder, and it is a 3D stack of different carbon layers. When it is exfoliated into a single carbon layer with sp2 hybridization, and these carbon atoms are basically arranged in a honeycomb like lattice, then it gives rise to the graphene. And graphene is a very important two-dimensional nanomaterial, and it has unparalleled electron mobility, which is a prerequisite for biosensors, because it gives excellent charge transfer capabilities, which help in the rapid detection of the analytes of interest. this. Then we have the graphene oxide. Over here, we are basically adding oxidative moieties, which make this graphene more hydrophilic in nature so that it imparts dispersibility in the aqueous media, which is important for biological functionalizations and detection of biological molecules in aqueous media. Then we have the zero-dimensional quantum dots. Now, quantum dots have fluorescence-like properties. So they are very, very important when it comes to the 3D imaging of different types of different types of target analytes. So these are basically semiconductor nanocrystals which have tunable fluorescence for multiplex optical detection in case of nanobiosensors. Then we have cerium oxide, zinc oxide, titanium dioxide. Now, these have unique catalytic properties. For example, cerium oxide is one of the important type of metal oxide which has very unique catalytic properties because of the oxygen vacancies created in case of the cerium nanoparticles. So, these can be easily clubbed to different types of graphene sheets or two-dimensional metal dichalcogenides for their nanocomposite-like behavior, which is much better when we are using a single nanomaterial. So such type of nanocomposites are also important for the detection of the different types of biological targets. So now, there are other types of 2D materials which we are going to discuss one by one. So one of them is the transition metal dichalcogenides, as I was discussing in the previous slide. So this basically has a general formula which is the MX2, where M is basically W or the molybdenum, and X is sulfur, ceralium or terralium. So basically, very common examples of these transition metal dichalcogenides are the MOS2 and WS2. The MOS2 molecule has a sandwich-like structure where the central plane of the molybdenum atom is sandwiched between two hexagonal layers of sulfur atoms, and these are bonded to the molybdenum atoms by the they are attached to each other by means of non-covalent actions, which is the van der Waals forces. So these can be easily exfoliated into single sheets and into the 2D sheets. So why these are so important? This is because the transition metal dichalcogenides have active edge sites or the defective sites which help in the charge transfer in case of the biosensors. Basically, the basal plane of these metal dichalcogenides is inert in nature, and the edges, they have active edge sites, and these active edge sites are the ones which are imparting the unique properties of the transition metal dichalcogenide. Now let us discuss the another important 2D material, which is the hexagonal boron nitride. It is also commonly known as the white graphene. Now in the white graphene, it has very excellent dielectric properties, and it acts as an excellent dielectric substrate, which helps in the detection of the target analyte. This is because in case of the optical biosensors, this dielectric layer confines the lights to the surface, which generates an evanescent field. Now, this evanescent field basically it penetrates the target substance only up to 100 to 200 nanometers. So, this helps in detecting even a small change in the refractive index, which occurs whenever a molecular binding event occurs between the bioreceptor and the target ligand. Now, another important 2D material is the black phosphorus or the phosphorine. Now, it has a very tunable band gap and high charged carrier mobility. So, it helps in the sensitive electrochemical and optical detection of DNA and protein. This is because these black phosphorus or phosphorine have a direct band gap, which helps in the broad spectral absorption from the UV as well as infrared range of the electromagnetic spectrum, which is particularly important for sensing as well as imaging in the biological samples. Now, another important topic, another important 2D material are the amexines. Now, they are a family of 2D transition metal carbides, nitrides, or carbon and electrical conductivity. Electrical conductivity of 2D material like the graphene is compromised. So in such cases, we can. This is because they are known to beat the beat even
Excuse me, madam. Am I audible? Your voice is not coming. Dr. Deepika Yadav, your voice is not coming. Tulji, please call her and again let her join. Thank you. I'm sorry, there is some technical glitch with Dr. Deepshikha Yadav. She will be joining back. In the meantime, I'd like to discuss about this few of the new developments which we have done under the legal methodology. As you are all aware, we have one Jan Sunwai on every Tuesday at 4 p.m. Anyone who has any issue can discuss at 4 p.m. online. The link as well as the password is available on our website consumeraffairs.gov.in. Anyone can join and can discuss or inform the issue if yes. Any of the industry, consumer, anyone can join on every Tuesday at 4 p.m. online. Secondly, we are also asking that these gold and precious metals weighing machines which are used for selling of these gold and precious metals should be used of even 1 milligram accuracy, 1 milligram accuracy or the verification scale interval, so that the error cannot be more than 1.5 e, that means 1.5 milligram. In place of now, generally or at few places, the weighing machines of 10 milligram least counts are available, which we are going to replace very shortly. We are requesting all the concerned user to replace these 10 gram balances with 1 milligram balance. And thirdly, we have also implemented the government approved test center rules. We have received few applications opened in November, and we are finalizing these all applications very shortly. By the end of this month, all these applications will be decided, and in the month of April, we will again start seeking applications for approval of GATC. So with these are the few new developments on which we are working. Anyone who is interested to have this verification or OML approval or something new can always contact me or contact any of the legal methodology officer in the state or the central level for any support. I think Dr. Deepshikha Yadav, you are back.
Sir, can you
Yes,
hear
very
me?
well. Please continue.
I'm so
No issue,
sorry,
no
sir,
issue,
for
no issue.
the
Please continue.
technical
No
issue.
issue. Please continue.
Could you, sir, could you please let me know from which
Last,
slide I
last
was?
slide, last slide. Immediately we spoke to you. Yeah, this one only. Please continue. Thank you. Thank you very much. Please continue.
Let us discuss what are the different types of nanomaterials which are used for 2-dimensional nanomaterials which are used for the nanobiosensors. One of them is the transition metal dichalcogenides. It has a common formula which is MX2, where M stands for molybdenum or tungsten, and then we have the X2. The X stands for the sulfur, ceralium, or teralium molecules. One of the common type of transition metal dichalcogenides is the MOS2, molybdenum disulfide nanosheets, and then we have the tungsten disulfide nanosheets, which are commonly used. The common structure is like a sandwich, where the central plane of the molybdenum atoms is sandwiched between two hexagonal layers of the sulfur atoms, and the sulfur atoms are bonded to the molybdenum atoms by means of strong covalent bonds. However, these layers of sulfur atoms are bonded to each other by non-covalent interactions, which is the van der Waal interactions. So they can be easily exfoliated into 2T sheets, which is imparting their unique catalytic properties in the biosensors. Now, the basal plane of these 2D sheets is usually inert. However, the edges of these molecules are highly reactive in nature, and they are commonly known as the active edge sites, which impart their catalytic properties. Then we have the hexagonal boron nitride, which is often known as the white graphene. Now, white graphene has imparts excellent dielectric properties to the biosensor surface. This is because why it is so important in biosensing, because in optical biosensors, this dielectric layer, it confines the light to the surface and generates an evanescent field which is capable of penetrating the sample only up to the 100 to 200 nanometer range. And since this field is concentrated just 100 to 200 nanometer at the sample surface, it helps in getting concentrated at the binding site, where the binding event between the bioreceptor and target analyte takes place. So even a very small change in the refractive index as a result of this molecular binding event can be detected. Then we have the black phosphorus or the phosphorine, and they have tunable band gaps and high charge transfer capabilities, high charged electron mobility, which is offering the excellent electronic transfer in case of the biosensors. So they have a direct band gap. So it allows for the broad spectrum absorption from the UV to the infrared range of the electromagnetic spectrum, which is important for the imaging as well as sensing analysis, especially for the nucleic acid as well as protein detections. Now, over the years, these 2D materials have had significant advantages over other materials and have contributed towards the recent generation of biosensors, or the fourth generation of biosensors. For example, in the transition metal dichalcogenides, they have intrinsic catalytic properties. So they have been used instead of enzymes because they have stability issues. Since enzymes are proteins, so they have to be highly stable in nature, and the environmental impact should not hinder with their 3D structure. So instead of using the enzymes, now transition metal dichalcogenides are used in the fourth generation sensing of different different biological elements, instead of using enzymes because of their potential disadvantages like the low shelf life and other degradation possibilities. So which might lose, because of which they might lose their active sites, which are responsible for the detection purposes. So why they are so important? Because they have high mechanical strength, they are very stable in nature, they can be scaled up, and they are cost effective, which is paving the way for their widespread integration into next generation of nanobiosensors. Now, another important material is the carbon nanotubes. Now, carbon nanotubes or nanofibers, they have been distinguished into single wall carbon nanotubes as well as multi-wall carbon nanotubes, and they both exhibit remarkable electronic properties. They have very high electrical conductivity, which is crucial for biosensing applications. Now, in CNTs, there is chemical bonding, a very strong chemical bonding, which is the covalent bonding in the side walls as well as ends. Now, basically, these side walls and the ends can be easily functionalized using carboxy as well as amine functional groups by following the oxidative treatment, which is also making them dispersible and stable in aqueous biological medium. They can also have non-covalent functionalization, which involves the physical adsorption of the biomolecules through van der Waals forces, pi-pi stacking interactions, which breaks down their typical sp2 hybridization of the carbon layers. Now, their important advantages include the high charge transfer capabilities, and they can be seamlessly integrated with the electronic and microfluidic system, which helps in the multiplexing of the different target analytes. Now, however, they also suffer from certain disadvantages. This is because they are not very biocompatible, which is a prerequisite for biological applications. They impart oxidative damage to the cells. Because of their hydrophobicity, they can also lead to the cell membrane damage. They also lead to inflammatory reactions occurring in the body, as well as they may also lead to the granuloma formation. So such type of toxicity challenges are hindering their usage. However, still they have a variety of advantages, which I had already discussed. Now we have the another important nanomaterial, which is the quantum dots, which are fluorescent nanomaterials for biosensing. This is because they can be used in imaging purposes. Because of their fluorescent natures, they can go and bind to the target ligand and lead to the fluorescence, which can help in their detection. Now, different sizes of these quantum dots can be used for the simultaneous detection of multiple biomarkers in a single sample. They can also be easily biofunctionalized to target specific molecules in case of immunoassays and cellular imaging. Now, there have been advancements in terms of reducing the toxicity, like the cadmium-free and perovskite quantum dots, which help in the circumventing the biocompatibility as well as the toxicity concerns which we commonly have. Now, quantum dots also allow the covalent as well as non-covalent attachment because they can be easily modified with chemical moieties like the carboxyl and amine. In addition to it, they can be also PEGylated using the polyethylene glycol layer. They can be PEGylated, which helps in maintaining their integrity and helps in preventing them from losing their brightness and also preventing their clumping together. So they can be individually PEGylated, which prevents their clumping to each other, which is important for the imaging analysis and maintaining their brightness. Now, another important nanomaterial are the gold nanoparticles or the plasmonic biosensors. As I had discussed in the previous slides, that gold nanoparticles have an important phenomena which is known as the surface plasmon resonance, where the collective oscillation of the electrons on the metal surface when excited by the incident white light leads to electromagnetic field, which is important for the label-free detection of the different types of analyte. Now, what exactly is this label-free detection? Now, label-free biosensing is basically an analytical method which helps in the identification as well as quantification of molecules by measuring their intrinsic physical properties like the mass, refractive index, and charge without the need of any artificial labels like dyes or radioactive tags. So, gold nanoparticles are particularly important because they have different colors. They come in different colors. So, based on their colors, they can be easily identified by the naked eye. So, we do not need any kind of radioactive labels or tags or dyes for the visualization of the target analytes. So, now they have size-dependent properties because gold nanoparticles exhibit tunable optical properties with color, and as the color changes, the absorption peaks also change based on their custom size. So they range typically from 10 to 100 nanometers in size, and this helps particularly in the custom sensor design. Now, they can also be used for easy functionalization because gold nanoparticles have affinity for different types of functional groups like amines, thiols. So, all of these groups are an important part of the biomolecules like proteins and enzymes, antibodies, etc. So, they can be easily targeted for their functionalization. So, with antibodies, DNA, and they are also used in addition with surface enhanced Raman spectroscopy because of their characteristic feature of surface plasmon resonance. Now, compared to other nanomaterials like quantum dots, they have superior biocompatibility. So, the toxicity and biocompatibility issue can also be circumvented. They can also easily be synthesized, and their size can also be very easily tuned and controlled for a multitude of different applications. Now, another important material is the graphene-based nanobiosensors. I had already discussed what is the difference between graphite, graphene, and graphene oxide. So, graphene basically is a single layer of carbon which is latticed in a hexagonal lattice, and it has exceptional electronic properties because of its 2D nature. Now, this includes the high electron mobility at room temperature, making it an ideal candidate for the highly sensitive detection, which is important in the biosensing department. Now, there have been recent highlights and breakthroughs where graphene has been used in the detection up to even femtomolar levels. So, which is very important, because there are certain ligands or certain target moieties which are present in very low concentrations in the sample, in the biological media or biological samples, which cannot be detected by conventional methods. So graphene based nanobiosensors are coming to the rescue over here. Now, GFETs are another important type of biosensor over here, which are using the unique electrical conductance of the graphene. Now, graphene oxide, as I had already told you, that when graphene oxide is functionalized with oxygen moieties, then it is getting converted into graphene oxide. Now, graphene oxide is hydrophilic in nature and can be dispersed in aqueous media, biological media, but excuse me, but somehow its electrical conductivity is compromised. So for improving its electrical conductivity, it can be reduced back into another substance, which is the reduced graphene oxide, and this has very high electrical conductivity. However, over here the hydrophobicity is increased, so the dispersibility is compromised. So they can be used in the label-free as well as real-time detection of different nanoparticles of different biological samples, and the high surface area can be leveraged for this particular detection. Now, there are challenges like the susceptibility to oxidation and degradation. They can also aggregate, and the consistent reproducibility across batches is also a rampant challenge. Now, let us discuss what are the key nanobiosensor types and what are their transduction mechanisms. So, they can be divided into electrochemical sensors, optical sensors, piezoelectric sensors, and thermal sensors. As I had already discussed, that electrochemical type of sensors, they are detecting the target analyte based on the changes in the current as well as voltage and impedance whenever the analytes bind to the sensor surface. So there is a change in either the current or the voltage or impedance, which helps in their detection. Then there are optical sensors which can be developed using nanomaterials which have excellent optical properties like gold nanoparticles and quantum dots, which leverage their fluorescence as well as surface plasmon resonance and Raman scattering enhancements for the detection of the analytes. Now, quantum dots and gold nanoparticles are one of the popular type of nanomaterials used here, and they help in signal enhancement with minimal background interference. Then we have piezoelectric sensors. Piezoelectric sensors detect the target analyte on the basis of mass changes through the frequency shifts, where we are using the quartz crystal microbalances or the surface acoustic wave devices. We can use different types of nanomaterial coatings which enhance the surface area, dramatically improving the charge transfer capabilities and detection ranges of the biosensors. Then we have thermal biosensors, which of course detect the heat changes which occur as a result of the biological reaction or the biological binding event which occurs at the electrode interface. Now, these nanobiosensors can also be integrated into the microfluidic system for developing powerful lab-on-chip type of platforms for the point of care testing, which helps in the detection of target analytes at the bedside of the patients, which is very, very important. Now, these integrated devices automate the sample preparation, help in the analyte delivery, as well as signal detection in compact and portable formats. Now, another important one is the mechanical transduction through the cantilever and acoustic biosensors. They are broadly classified into three types, which is the micro cantilever sensors, quartz crystal microbalance, and surface acoustic wave devices. Now, micro cantilever sensors are based on the molecular binding event detection through the mechanical deflection. Whenever there is a target binding occurring at the electrode interface, the functionalized cantilever, it bends, so it provides a quantifiable signal for detection. Now, this approach, why it is so important? Because it helps in the label-free detection without imparting the toxicity element in the biosensors. Then we have the quartz crystal microbalance, whereas the crucium or the quartz crystal is used, which vibrates at a specific resonant frequency. Whenever biomolecules get absorbed onto this crystal, there is a change in the mass, and this causes a change in the frequency. So this particular mass sensitive detection mechanism helps in the real time detection of the biological interactions. Especially cell adhesion as well as protein adsorption type of studies are quite popular using these quad crystal microbalance. Then we have the SAW sensors. Now they operate by the generation and as well as detection of acoustic waves at the surface of the piezoelectric surface. Whenever the target molecule they get absorbed onto the surface, it leads to the change in the mass. And whenever there is a change in the mass, the velocity as well as amplitude of the propagating wave gets altered. Now, all of these changes can be detected by these SAW devices, which help in the highly sensitive detection of the target analyte into a compact biosensing platform as well, which helps in the development of point of care devices as well. Now, all of these techniques are highly sensitive and also provide label-free detection, but they also suffer from temperature sensitivity baseline drift, and because of their complex nature, they require sophisticated instrumentation, which is also a possible challenge. Now, however, there is ongoing research which is taking place where they are being integrated along with microfluidics for the more robust as well as portable device development. Then we have the optical transduction, which is based on the fluorescence and absorbance based detection. Over here, the non-radiative radiative energy transfer occurs between the donor as well as the acceptor fluorophores, which is important for the target detection. Then we have time resolved fluorescence, which distinguishes the target fluorescence from background noise by measuring the decay kinetics, which helps in the significant improvement in the signal clarity. Now, then we have the absorbance as well as colorimetry based detection, where we can help detect the changes in the light absorption as well as any type of visible color shift which occurs as a result of the biomolecular binding event. Then we have the Raman as well as the surface enhanced Raman spectroscopy, which helps in the detection of in the detection of target analytes using the optical nanoparticles for the significant enhancements and amplification of the signals, which is responsible for the high sensitivity and ultra sensitive detection down to femtomolar ranges as well. Now they have advantages like high specificity and sensitivity, and they can also be multiplexed, and they have non-invasive and real-time measurement capabilities as well. They can also be integrated along with optical fibers for the enhanced sensitivity. But there are also challenges that the photo bleaching of the fluorophores can occur. Also, there are certain biological molecules which also give fluorescent signals, so they can suffer from interference from those. And then light scattering, because the blood or the human biological samples, biological media is highly scattering in nature, which is responsible for these light scattering. Now, since the detection technique involves sophisticated instrumentation, it leads to the generation of complex data, which requires skilled personnel for the advanced data interpretation. Then we have the electrochemical transduction as well as their principles and performance. Over here, the electrochemical biosensors basically convert the biological recognition event into measurable electrical signal in terms of the current, voltage, or the impedance response. As we had discussed in my previous lecture also, that there are different types of electrochemical biosensors like the amperometric, voltammetric, as well as impedance spectroscopy, wherein there are different biorecognition event leads to the change in the current as well as the potential and the impedance. So whenever there is a binding event which occurs at the electrode surface, there is a change in these the electrical properties, which is harnessed in case of these electrochemical biosensors. So the amperometric detection basically measures the current generated, which occurs as a result of the binding event. Then the voltammetric technique involves the sweeping of potential from one point to another point and then back to the same point, which is the type of cyclic voltammetry. Then we have the differential pulse voltammetry, which helps in enhancing the sensitivity even for trace analysis. Trace analysis in the sense that the concentrations of the target analytes is significantly low, so DPV is a very sensitive technique for detecting them. Then we have square wave voltammetry, which offers rapid analysis having very high signal to noise ratios. Then we have the electrochemical impedance spectroscopy, which is another important technique wherein the change, whenever we are depositing the electrode surface with any kind of biomolecule or any kind of bioreceptor, there is a change in the impedance values. Because whenever we are labeling the surface with any type of biomolecules are having low conductivity. So whenever they get deposited, there is a change in the conductance. There is a resistance to the flow of the electric charge, which can be leveraged by this technique. Now, there are advantages as well as challenges associated with this electrochemical technique as well, like they have high sensitivity and specificity and help in the real-time monitoring, and they can be easily miniaturized and are quite low cost and portable. However, they suffer from the electrode fouling properties as well as the background noise and interference, because biological molecules, the biological media is a complex matrix. So it leads to the matrix effect, which wherein the non-target molecule also lead to the signal, and which may lead to the false positives as well. And also it suffers from low reproducibility. Now, there are measurement challenges which are there in nanobiosensor, that is sensitivity, selectivity, and speed. Now, whenever we are trying to achieve sub-picomolar detection limits, it demands overcoming intrinsic electronic noise and as well as thermal fluctuations. Like I told you that whenever we are using biological media, it is a very complex matrix. So there are non-specific binding also which take place. Therefore, reducing this background noise and reducing the interference from non-target molecule is a long-standing hurdle in nanobiosensing. Then we have response time constraints. The rapid analyte sensor interaction requires very efficient mass transport at the nanoscale. Now, innovative solutions include nanostructuring surface to create connective flow patterns, using magnetic nanoparticles, and designing hierarchical porous architecture that maximize the binding event at the binding site. Now, we also have selectivity hurdles. The matrix effect is a very big hurdle standing in the way of the nanobiosensors because of the different interference from other non-specific target, non-target analytes or non-specific analytes, which leads to the false positive results. For this purpose, nowadays molecularly imprinted polymers are being used which have shapes which are complementary to the shapes of the target analytes. Now these are being designed for this purpose for reducing the non-specific interactions. The signal to noise ratio may also be low in certain cases. Then we have the matrix effects, where real world samples contain interfering substances which may interfere with the detection. Then we have reproducibility. Whenever we are fabricating at the nanoscales, reproducibility is an important factor, because even small type of variations may lead to large errors. Therefore, the precise development of nanoscale materials is quite a challenge with high reproducibility. Then we have response time and kinetic limitations in nanobiosensors. Whenever we the key kinetic challenges include the diffusion limits, binding kinetics, and signal transduction. In case of diffusion limit, the analyte mass transport to the sensor surface often is dictating the speed of the response. So basically, whenever the target analyte is moving from the bulk to the sensor surface, there are different types of diffusion limits which are dictating the overall speed of the response of the fabricated sensor. Then the association as well as dissociation kinetics of the different types of bioreceptor also affect the overall response time. Then we have the signal transduction. The inherent time constants for converting a binding event into a measurable electrical signal also leads to the delays in the detection times. Then we have optimization strategy. Microfluidic, whenever we are taking the microfluidic integration into account, the precise control is important, which is helpful in enhancing the mass transport and also reducing the diffusion distances. Then whenever we are using these nanobiosensors, if we are introducing active stirring or flowing samples, this also significantly enhances the response time. We also need to balance the kinetic measurements with slower, more sensitive equilibrium approaches for the effective applications. Then we have the sensitivity versus selectivity, the fundamental trade-off. Now, quantitative metrics. In my previous lecture, I had discussed about the limit of detection, limit of quantification, and the dynamic range. Now, all of these parameters are important for defining the sensing capability of the nanobiosensors.
तो इसमें एसएमएस कर रहा था, मेल आईडी तो मेल में कर देंगे, तो वो कोटेशन वगैरह सब दे दूंगा आपको.
Excuse me. Hello. Hello.
One much free.
Sir, can I continue?
If you wish to, you can continue for 5-10 more minutes.
Okay, sir. So we also need to minimize the cross reactivity in case of the matrix effect. I'll just quickly glance through the remaining slides. Now, we also need to overcome the different types of challenges in the nanotechnology, like the nanoscale confinement. Now, restricting the sensing volume down to the trace concentrations is quite challenging. We need to enhance the signals and minimize the background. For this purpose, we can use the nanopillar arrays or nanopills, which help in concentrating the analytes and minimizing the background. Then we have the nanopore blockade sensors, where we can detect single molecule protein by monitoring the ionic current. Then we have multiplexed arrays, where different types of multiple analytes can be detected in high density sensors. Then we have when we use robotic liquid handling and automated data analysis, it reduces the human error efficiently and helps in the high throughput screenings. Then we have the biorecognition elements. They can be antibodies, aptamers, or enzymes. Antibodies are highly specific immune molecules which are helpful in detecting the target analyte because they are highly specific in nature. Then aptamers are synthetic nucleic acids which are being used nowadays popularly instead of antibodies because of the low shelf life and other issues related to the antibodies, and because antibodies are also very costly molecules. So we can also use aptamers instead of them because they are capable of detecting the different types of proteins. Then we have enzymes which are biological catalysts which can also help in the detection of different types of target molecules. Now this particular bar chart helps in differentiating the specificity, scalability, stability, cost effectiveness, and regeneration of these different types. Over here we can see that the aptamers have a strong performance in terms of the scalability, stability, and cost, while enzymes excel in the specificity. Beyond these primary types, we can also use other types of biorecognition elements like bioproteins, protein fragments, nucleic acid probes, which can also be used for enhancing the signal and detecting the target analytes down to the sub-picomolar ranges. Then we have different types of signal amplification strategies like the enzymatic amplification, where we can use enzymes like hostidic peroxidase and alkaline phosphatase, which help in target binding because they produce a large number of detectable products like colorimetric, emulsient, or fluorescent. Then we have catalytic nanoparticles like the common gold nanoparticles, which help in enhancing the signals and developing fourth generation of biosensors, where we are replacing the enzyme using these catalytic nanoparticles. Then we have isothermal rolling circle amplification, which uses circular DNA for enhancing the signals. Nanobiosensors can also be used for the detection of different types of infectious diseases and for epidemiology analysis, because they can be used for the timely detection of antimicrobial resistance, so that we can adjust the dosage and we can know how much of drugs we need to administer in order to prevent the phenomena of antimicrobial resistance. Then nanobiosensors can also be used for dental health as well as oral diseases, where they can be used for the detection of oral cancer biomarkers down to the very low sub-picomolar ranges. Nanobiosensors are also being used for vaccine development because they can be used for studying the real-time analysis of antibody and T cell responses. Because whenever we are able to detect the antibody, we can help in providing comprehensive insights for for understanding the immune system's reaction to the vaccine, which helps in their rapid development, which is responsible for personalized as well as cost-effective vaccination programs. Then we have the multiplexing and multi-analyte detection platforms. Using these nanomaterials, we can develop systems wherein we can detect the multitude of analytes in a single array, which is helpful in detecting different types of diseases and helpful in detecting the comprehensive biomarker profiling and identification of intricate disease signature and reduce sample volume. However, it also helps in the early cancer diagnostics and prognosis and simultaneous detection of multiple biomarkers. Nanobiosensors can also be used for metabolic disorder management because glucose biosensors are a common type of nanobiosensors, and nowadays non-invasive glucose biosensors are also being developed, wherein we can help in the development for the monitoring of glucose as well as for insulin dosage monitoring, and also providing the real-time data for optimizing insulin dosage and helping in the systematic glycemic control, preventing the diabetes-related complications. We can also use them for viral infection detection like the RSV, influenza virus, HIV, hepatitis C viral loads, which can help in the prevention as well as identification of prevention of such diseases and rapidly identifying novel and re-emerging viral strains, which can lead to the different types of diseases. Nanobiosensors are commonly used for pregnancy detection, like the common pregnancy detection kit, which helps in the detection of HCG hormone, human choreographic hormone, and progesterone and estrogen for the early confirmation and monitoring of the pregnancy. It can also be used for personalized medicine and pharmacogenomics profiling as well as real-time drug monitoring and helping in the enhanced patient outcomes. Nanobiosensors, as I told you, can also be used for the antibiotic resistance detection, which helps in the contributing towards the inappropriate antibiotic usage and significant cost cuttings. They can also be used in cancer biomarker detection for circulating tumor cells, like circulating tumor DNA, exosome-based diagnostics, which are basically extracellular vesicles, which are responsible for the crosstalk which occurs between different types of cells for the transport of different important materials. Then we have the future trends and next generation of biosensors. We need to miniaturize and develop flexible electronics for ultra thin flexible sensors. Then we also need to integrate the artificial intelligence algorithms for processing the large and complex sensor data. And the green chemistry helps in the development of different types of nanomaterials using extracts, enzymes, or microwave assisted synthesis. Then we also need to to assess the biomaterials for their biocompatibility and initial as well as long term, where initially we can detect their toxicity using the in vitro cytotoxicity and in vivo model. In vitro is this lab scale testing on on cell lines, and in vivo are the lab scale testing in the animal models. Then we can also test for the long-term biological impact by studying the biodegradation, organ accumulation, and the immune response, or the genotoxicity which is induced. We also need to standardize as well as do the quality assurance using the different types of ISO standard. We need to comply to those for nanomaterial characterization, and we also need to use the biological reference materials certified standards for the calibration. We also need to adhere to good manufacturing practices for traceability, for full documentation from the raw material to finished product. Then artificial intelligence and machine learning in nanobiosensor data analysis, because nanobiosensor generates large and complex data for which artificial intelligence and machine learning comes into picture for the real-time decision support and regulatory compliance. We are also using the wearable and implantable nanobiosensors for the next generation frontier, where flexible materials can be used for the subcutaneous implants, and then they can also be connected to the wireless and Internet of Things application, so that the large data which is generated can be easily harnessed and can reach the medical professional on time. Then we have the performance validation, where we need to validate the performance using the limit of detection, the specificity and sensitivity, the accuracy and the dynamic as well as linear range, selectivity testing, stability testing, batch testing, and statistical analysis. There are different types of characterization techniques for the nanomaterials, which are the transmission electron microscopy, scanning electron microscopy, atomic force microscopy, which is responsible for detecting their size as well as surface morphology and mechanical properties. Then we have the XPS technique, which is helpful in determining the elemental composition and the chemical states, and the Raman spectroscopy for molecular vibrations, and UV visible spectroscopy for identifying their unique optical properties. Then we have microfluidic integration and lab-on-chip devices, because we need to multiplex the nanobiosensor for detecting different types of analytes on a single platform. The biorecognition element can be immobilized using different types of technique, that is the covalent through strong bond, through strong stable bond, via the click chemistry or the carbonamide coupling, the affinity base, wherein highly specific interactions like biotin, streptavidin, histidine tag can be used, and then they can also be physically absorbed through electrostatic interactions. We can also use the self-assembled monolayers for the precise control of the film thickness and surface chemistry. Then we can use polymer coatings as well as hydrogels. Nanobiosensors can be developed using bottom-up synthesis using the CVD, wet chemical method, and sol gel methods, which are common. Then we can also use the electrochemical deposition and self-assembly. The nanobiosensors, the top-down approaches include the photolithography, focused ion beam, and reactive ion etching. These approaches offer high precision, reproducibility, and scalability for microelectronic manufacturing and batch processing. However, they are high cost and have resolution type of limits. Now, the visualizing the impact from lab to market and beyond. Now, we have improved the detection fold increase in sensitivity from projected micromolar to down to attomolar concentration from 2000 to 2030 with over past 2 decades. Now the market value is about $500 billion, and it is very important because it is giving a vast market value to these nanobiosensors. The test time is average from sample to result is within 15 minutes. Now, there have been dramatic improvements in the detection limits from 2000s to 2025. We have been able to achieve down to attomolar levels because of the integration of the different types of nanomaterial imparting their unique physicochemical and optical properties. there are different types of challenges in clinical translation, like regulatory complexity and approval timelines, which significantly delay the process. The clinical validation is also required. The intellectual property challenges, manufacturing scale up, competitive landscape, all of these are quite challenging in the clinical translation. However, there are certain strategies which can help in overcoming, like the effective early engagement with regulatory bodies, strategic partnership for clinical development, and demonstrating clear economic and health benefits. Now, conclusion: the promise and path forward for nanobiosensor. Now, nanobiosensor field stands at a pivotal inflection point where laboratory demonstrations are transitioning to commercial products, and standardizing these fabrication protocols will ensure reproducibility. Crosstalk with the regulatory frameworks will help in the speeding up the approval process. And then we need to manufacture innovation, reducing production costs, and clinical validation should also be done in diverse patient population. The future of medicine lies in our ability to detect disease at its molecular origins, and nanobiosensors are the instruments that will make this vision a reality. These are the intellectual property and patient then these are the regulatory pathways where the FDA approval is important, and the global nanobiosensor market, which helps in forecasting the size and the growth projections. Thank you so much for your attention.
Thank you very much, ma'am. Thank you for your brief and wonderful presentation. I'd like to request you to kindly remove this PPT slide.
Yeah, sure, sir. Yes.
RSL Ahmedabad, will you please take photographs? I would like to request all the participants to kindly switch on their video. And I would like to request you to kindly share me your PPT so that I will put it on our website. Atulji,